Fill the sheath tank by removing the lid, replenishing the sheath and replacing. Empty the waste by removing the lid, discarding the waste safely. Add in a disinfectant such as bleach as needed and replace the lid. Press the button on the front of the machine to turn it on and also launch the PC. Open the ID7000 software. If the machine hasn't been on for a few hours, the machine will automatically prompt you to prime the system. Run this program, which takes approximately 10 minutes. Start the daily QC and follow the prompts to place a tube of align check beads in the additional sample holder. After this, you may wish to run the performance QC as well, but it is not necessary for the functioning of the machine. Once the daily QC is completed, you are ready to begin your experiment. I'm going to show you how to set up and run a new experiment from scratch, but there are a number of different ways you can start this experiment. If you've run the same samples before, you can open a template, or you can click on a previous experiment to use this as a template. When doing something completely new, you can use the Experiment Designer, which takes you through fluorophores and markers, and can be an efficient way to set up, especially if you're doing a panel with lots of colours. In this video, I will use a blank template, but whichever way you begin an experiment, the principles are the same. So today I have my samples in standard fax tubes, so I will choose Experiment Template, Blank Template, 24 tube rack. You've got the option to change the name of your experiment and other minor details. Once you're happy, click Create Experiment at the bottom. Here on the left you can see we have our tube rack layout. The first thing that we need to do is to create the sample group. The sample group you can think of as a protocol. It will contain all the instrument settings, the unmixing settings, including the fluorophores, markers, as well as the worksheets. So we can highlight some of the tubes that we're going to use and move them to a new sample group. Once created, I like to rename the sample group as if you were naming a protocol or template on any other machine. So this refers to the settings that you're using rather than the samples that you're going to run. Right-click on the sample group, choose Properties, and I'm going to name my sample group PBMC's 4 colours. This machine has 147 fluorescent detectors, so we certainly don't want to set all of those from scratch. You're provided with some default settings which work well for many PBMCs of human and mouse, be they live or fixed. As we are running PBMCs, I'm going to load these settings. On the top ribbon under Acquisition, click Import Instrument Settings and Import from File. Browse to where the settings are saved, and I'm going to choose the default settings here. Press OK and import, and now all the settings for all the detectors are loaded. Remember, we're creating this experiment from scratch here, but if you were starting from a template or a previous experiment, then all of these settings would already be ready for you. As you use the machine more often, you may build up a bank of these instrument settings for different cell types. By default, the ID7000 saves all the data from all the detectors, but to see specific fluorophores, you will have to tell it what you're interested in. So even if you're just doing a one-colour experiment, let's say you just wanted to measure GFP in your cells, you still need to tell the software that it's GFP that you're interested in. Fortunately, the software has an extensive database of fluorophores, so this is easy to do. To do this, we need to go into the unmixing settings, click unmixing, and then unmixing settings. And the first job is to add our fluorophores. Simply type in the exact fluorophore that you are using. Chances are it'll be in the database. So here I'm going to add Brilliant Violet 421. So I type in BV421 and it appears. At this point, I highly recommend typing in the marker name. My marker is CD4. You can then repeat that for all of your fluorophores in the experiment. Here we have 4 colours. Note, if your fluorophore isn't in the database, then type its name in, click Add, and then choose the lasers and detectors that you want to use. Don't worry too much about the exact spectra because the machine will extract that later. Now we can create some plots. We already have a spectral ribbon and a plot of forward and side scatter. So at this stage, let's just add in a histogram for each fluorophore that we are working with. Click on Workspace Tools, and you can see that you can create different types of plot. Here, I'm going to create histograms. Click the histogram button, and then on the x-axis, select your fluorophore. Do that for all of the colours you're using. Open the door and place your tube rack in the machine with position A1, the front left. Pull the metal pillion near the D6 corner to allow the rack to drop into position and close the door. Here my samples are in the 24 tube rack, but you might have a 96 or a 384 well plate. Place them in the same direction with A1 to the front left. So now we can name the samples by using the list and clicking on a sample and typing the name in the box. Or use the layout editor where you can click on each well and type its name and any other extra information. It's worth noting that you can import and export these layouts, which is particularly useful for large experiments and 96 well plates. To check the instrument settings are appropriate, you'll need to analyse some unstained cells and some fully stained cells. Click on the tube or well that contains the unstained cells, right-click and choose Set Current Position. Click Instrument Settings, and on the Flow Control tab, reduce the flow control, which is the speed at which the machine processes the sample. Set it to between 1 and 3. 1 is 33 microliters per minute, 3 is 70 microliters per minute. Click Load and acknowledge the machine is cooling. We're now ready to look at this sample, and I'm going to do that by pressing the Preview button. We use this button when we are setting up the machine. First, I'm going to check the forward and side scatter. You may need to zoom in to get a clearer picture. To do this, right-click and press Zoom, then draw a rectangle around the region of interest, going from top right to bottom left. The cells that we are interested in here are these lymphocytes. So I will right-click and press Insert Gate, and I will choose here the ellipse tool to draw around these cells. We can now apply this gate to all of the plots. These cells are unlabeled, so we're using them to check each fluorophore and to see where the negative cells appear. These cells are all appearing fairly low down the scale of each histogram, and we want this to be typically between 10 and 1,000 units. So in this case, we don't need to change the instrument settings. If we were running larger cells, such as epithelial cells, stem cells, or dendritic cells, you would probably find them higher up on the scale, perhaps at 10,000 or 100,000 units, in which case you would need to reduce the voltage of the detectors to allow room for the stained cells. If you were running bacteria or extracellular vesicles or other small particles, you would find them low on the scale, so you'd want to increase the voltage to make sure that you had good sensitivity. To adjust detector voltage, click Instrument Settings and Detectors and Threshold. You will see that you can adjust all the detectors. Because there are so many detectors on this machine though, we rarely change individual ones. Rather, we change all the detectors for a given laser at once. If the cells were too high up, we would identify the laser being used to excite the fluorophore and then reduce the PMT voltage percentage for the detectors of that laser. Here the unstained cells are fitting nicely on the lower end of the scale, so I'm not going to make any changes, and I'm going to press Stop for this sample. So next we're going to preview a fully stained samples. We haven't set up any unmixing yet, so we just want to use this sample to make sure that none of the stains are too bright. We don't want them to go off the top of the scale and saturate the detectors. Once you're happy with your unstained and your fully stained or within the detector range, you can keep those instrument settings and proceed to set up the remainder of your experiment. To allow us to do spectral unmixing, you need a set of single colour controls, which are beads or cells stained with a single antibody or single reagent. Here my samples are compensation beads, so I'm going to preview the first sample so I can create a gate on the forward and side scatter plot to identify those beads. Go into the stopping conditions and set it so 10,000 beads trigger the stop. Then highlight all the bead controls, right click and choose Add to Auto Acquisition Target. This programs the machine to run all these tubes sequentially. Press the Auto Acquire button. The machine will process all the single colour controls that I've got on beads here. If you also have some single colours on cells, set the machine up and run those single colours that are on cells until you have finished all your single colour controls. Now we want to set the stopping conditions to acquire the desired numbers of cells. It's a good idea to set a time cutoff as well to stop the tube running dry. If the flow control is set to 10, this will use 200 microliters per minute. So a setting of 2 minutes for a 500 microliter sample will stop the machine running dry. Here I'm going to choose the stopping conditions with gate A, and we can set the desired number of cells to acquire. So let's say 50,000 cells. Once you've set your stopping conditions, you're ready to run the samples. Simply press Auto Acquire. Choose a single colour control and find the histogram for the fluorophore of interest. Draw a line gate on the histogram. It doesn't matter exactly where you place it, just as long as it's on a part of the positively stained beads. Check the spectral ribbon by selecting the gate that you've just drawn. You can now see the profile. Here we're looking at brilliant violet 570. So we can see on the spectral ribbon under the 405 nanometer laser, the violet laser, we can see a peak which approximates to 570 nanometers. This is a nice clean spectra and will work nicely for our unmixing. Right click on the linear gate that you've just created and assign this as the positive gate here for brilliant violet 570. Move to the next single colour control sample and repeat this process for each fluorophore, inserting a linear gate, checking the spectra, here we've got brilliant violet 421, and assigning it as a positive gate. The next sample is brilliant violet 650. The last sample is PE E-fluor 610. We can see here we've got a nice spectra. We don't want outliers. If there are outliers, you may wish to adjust your gate. We also need to set a negative gate on some unstained sample. So we need some unstained beads in our case. You may have a separate tube where no fluorophore has been added, but in this case, it's clear that in this sample, there is a negative population which are completely unstained. So we draw around this negative population, again using a linear gate, and if we select this gate on the spectral ribbon, this is a typical profile for compensation beads. We can now right-click on this linear gate and set this as a universal negative control. As all our samples are of the same bead type, we can choose the universal negative control. But if you've got samples where the single colour controls are on mixed, say cells or compensation beads, you can set multiple negative controls for individual fluorophores. To calculate spectral unmixing, click Unmixing and Unmixing Settings. You can see all the fluorophores have now got gates for the positive and negative. We can click Calculate, and at this point a warning comes up that there are some saturated events. Just press Continue. The machine will now extract the spectra for all of our fluorophores. So here we go. We can click on an individual fluorophore and see the spectra that has been calculated. Here we've got brilliant violet 421, and you can see that we've got a nice peak in the first few channels of the violet laser, a peak in the UV laser. So this is a nice spectra for brilliant violet 421. Critically, we do not want a spectra that goes below the 0 value. If you do have this, you may need to go back and adjust your unmixing settings. Often it's a problem with your negative control, so try regating your negative. Once you are happy with all your spectra, press Apply, and the spectral unmixing will be applied to your data. So for this sample, this is CD28 Brilliant Violet 650, and you can see that we've got a clear positive peak for this fluorophore, but all the others are now back down to baseline. Let's look at our fully stained sample. First, make sure that all your histograms are gated on your cells gate, which is gate A here. So we can see the data. It looks nice. We've got several peaks. We can inspect it further by creating further histograms and density plots. So let's have a look. Let's say we choose CD4 on the x-axis and CD8 on the y-axis. We can see the data now. Often you need to rescale the data. A quick way to do this is to click on the plot and then press the Auto Adjust XY button. There we go. Now we can see we've got nicely resolved populations. Our data is looking good. To further inspect all the unmixing, you can click Unmixing and Unmixing Viewer. This is a really nice way to check for any minor problems with the unmixing. At first, you may need to highlight the plots and press Auto Adjust XY. And then at the top left, you can select any given fluorophore and it will display this fluorophore on the x-axis against all the other fluorophores on the y-axis. You can press Next and scroll through all the fluorophores all the time looking for any problems with the unmixing. Make sure that you're gated on your cells. Here it's gate A. If you have a viability stain, you may wish to gate on the live cells. If you have a small problem, you can use the adjuster to correct it. Just click the adjuster button on and you will see a diagonal line appears on all of the plots. So here, perhaps there's a minor problem with CD8 PE eFluor 610. Perhaps it's been slightly overcompensated into CD4 Brilliant Violet 421. So if you wanted to change this, you can simply click on the positively stained population and drag it. You will see the population move slightly. Adjust these as you want, and once you're happy, click Close, and if you've made any changes, the software will prompt you to save it. You can either overwrite the unmixing, or you can choose to rename the unmixing. Right-click on the sample group and choose Export to FCS. Here you can choose the save path, and I would also recommend removing height and width for the fluorescence channels to reduce the file size. The resultant FCS files can then be opened by any third-party software and should contain perfectly unmixed data ready to be analysed. When turning the machine off, first remove the sample rack or any plates, close the door, click Cytometer, Hardware Shutdown and choose bleach cleaning and rinse. Load a tube of bleach and a tube of water as directed by the software wizard. Press start. Once the cleaning is finished, press shutdown and you will hear the machine power off.